Hello everyone, I am Sunil Kumar Sahu from BGI Shenzhen. I am very glad to share my research on the chromosome scale genomes of two Dipterocarpaceae species which provided insights into fragrant oleoresin biosynthesis and hardwood formation. If you have watched a documentary on or been to a tropical forest, it is very likely that you have seen at least one species of Dipterocarp. Biologically, Dipterocarps are quite fascinating. Wherever they occur, they form the backbone of the forest ecosystem. You might have seen this viral video on social media regarding the unique seed dispersal mechanism of sal tree. The Dipterocarp family belong to the order of malveals and consist of 16 genera and around 700 species, which are mainly distributed in the tropical lowland wet rainforest area as shown in the map here. The Dipterocarpaceae species serve as the principal source of fragrant oleoresin, which are present primarily in the tree trunk. They are also famous for the high quality timber, and the bioactive components are used for the preparation of traditional Chinese medicine. And thus, we can confidently say that Dipterocarpaceae are of high economic value, and money does grow on trees. Considering the increasing demand in the market for the resin and the high quality timber of different Dipterocarp species, there is also declining population. Therefore, it is important to interrogate the genomic background to explore the genomic features of Dipterocarp species. Therefore, the present study was designed to generate the chromosome scale genome assembly of Dipterocarpus turbinatus and Hopea hainanensis to explore the evolution and differentiation mechanism of oleoresin biosynthesis and hardwood formation. Coming to the methods, both the species were collected from Ruili Botanical Garden of Yunnan province of China. We employed both short read, nanopore, and high-C to generate the chromosome-level genome assembly. And using the multiomics data and comparative genomics approach, we tried to gain insight into their unique aroma and wood formation. And coming to the results, as you can see from this table, we have very high N50 values with more than 90% BUSCO, and around 40,000 and 36,000 genes were identified in each species. And also the LTR assembly index of more than 210 shows the reference quality genome. And based on the high C mapping, we were able to identify 11 and 7 pseudochromosomes in each species. And the phylogenetic analysis showed that the common ancestor of the Dipterocarp experienced a shared whole genome duplication event, and around 20,000 gene family were Dipterocarpaceae specific, while 11,000 gene families were shared among the malveals family. We also detected 2 is to 2 is to 1 syntenic relationship between D. turbinatus, Vitis vinifera, and Hopea hainanensis. Among the 5 duplication types, we were able to identify that whole genome duplication event mainly contributed to the highest proportion of expansion of the gene family, mainly related to the terpene and phenylpropanoid biosynthesis, which likely contribute to the characteristic fragrance aroma of these two species. And then we further focused on their gene expression profile and found that majority of the genes were exclusively expressed in the wood tissue compared to the leaf tissue. Likewise, SABATH, which is also very important for the phenylpropanoid and benzenoid biosynthesis, were also expanded when highly expressed in these two species. And then we further looked into the evolution of terpene biosynthesis and found that 8 special TPSA genes in the Dipterocarpaceae, which are not found in any other species, And interestingly, we found several TPS gene clusters in like chromosome 4, 8, 7, and 1, which suggests that these genes underwent a recent tandem duplication event. Then we also focus on the evolution of the terpene biosynthesis and its regulatory gene. And one gene that is BHD acyltransferase was highly expanded in this species, and we found that several genes in this species also showed very high expression in the stem tissue compared to the leaf, similar to other pathway. Likewise, when we looked into the cellulose synthase and cellulose synthase-like genes, we found very high expansion in both the tree species. And coming to the lactase and peroxidase, which is involved in the lignin metabolism, they also showed very high expansion and expression, gene expression activity in the stem compared to the leaf tissue. Then we looked into the gene regulatory networks of these two species and found that AP2 and EREBP and BHLS transcription factor played very close association with the fragrance and wood biosynthesis. And coming to the phytohormone, we found that salicylic, jasmonic acid, and abscisic acid were highly accumulated in the stem tissues of Hopea hainanensis, or on the other hand, D-turbinatus displayed fourfold higher gibberellic acid accumulation in the stem than leaves. Finally, in summary, we were able to generate a very high quality genome assembly of two representative Dipterocarpaceae species, and we were able to identify key insight into the fragrant oleoresin biosynthesis and hardwood formation using the multi-omics data. This research was recently published in the Plant Biotechnology Journal. And finally, I would like to thank our director, Dr. Liu Han, Wang Sibo, and Liang Hongping for their nice contribution in this work, and our funding from National Key R&D Program of China, Major Science and Technology Projects of Yunnan Province, and Shenzhen Municipal Government. Thank you. Thank you so much for your patience listening. I would be very happy to take your questions. Thank you.